In this video, we will discuss about SARS-CoV-2 and vaccine pathway with the help of figure that has been presented in one of the papers. The reference of that paper will be provided in the description. So this is the figure in this. Yeah. So let us first understand how the SARS-CoV-2 virus actually infect the cells and how it uh, take the advantage of the cell machinery. So this is the SARS-CoV-2 virus, and this green part is the ACE2 receptor. So and the blue part is the spike protein. The spike protein of the SARS-CoV-2 virus binds with the ACE2 receptor that helps, and this S1 and S2 both, S1 helps in binding with the ACE2, and S2 helps in then fusion to the membrane and then entered, or we can say move in the cytosol of the cells. And when the virus is in the cytosol of the cells, it release its viral genome, and then translation of the viral protein occurs, and code translation, you can say, while translation during code translation of the viral protein, it enters into the endoplasmic reticulum for the post translation modification, that is N-glycosylation. And when it moves further in the process in the Golgi apparatus, the O-glycosylation is also occurs. So when it moves to the when the translation process completed and the N-glycosylation started in the endoplasmic reticulum, the spike protein formation occurs, and that is represented by this S symbol, and these two different colors represent S1 and S2 site, and this M represent the membrane part of the virus, E represent the envelope part of the virus, N is the nucleocapsid. So the spike protein is the process, the co-translation process of spike protein occurs via endoplasmic reticulum, then it moves to the Golgi, then to the via lysosome pathway it moves out. So when the spike protein is translated in the endoplasmic reticulum, the N-glycosylation also begins at the signature sites, that is NXT, where N is asparagine, X is any amino acid except proline, and S and T are serine and threonine. So after this N-glycosylation started, or the pre-assembled glycan attached to this signature site of the N-glycan, it then moved to the iric lumen, that is endoplasmic reticulum Golgi intermediate compartment, where it encounters with the uh, budding 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 virus, or you can say that nucleocapsid. And after that, after it moves from the iric pathway, the viral genome is covered with the nucleocapsid, and in the cis part of the Golgi, the manostreaming and the complex initiation occurs, and when it moves to the medial part, the further elongation and further modification of the glycans occurs. At the same time, O-glycosylation also begins, and when it moves to the trans-Golgi region, their fucosylation or silation occurs, and at the same time, this spike protein get cleaved by host furin protease, and even after cleaving of the spike protein by host furin protease into S1 and S2, S1 and S2 still are covalently attached to each other, and this affects the degree of further post-translation modification or further glycosylation. After that, it moves because of the signature of the signature present on the virus. It moves to the lysosome. It moves to the lysosome, then to the extracellular matrix to affect another cells. And when there is another DNA, and now let's understand how this DNA or RNA kind of RNA type of vaccine works. So this DNA RNA vaccines, it also it is uh, it also it is directly injected to the cells, or it is also use some kind of receptor binding domain that might be ACE2 or uh, some other receptor binding domain, or can be directly injected. A plasmid can be directly injected into the cell, where the release of DNA occurs, and the here also translation of spike protein occurs in the endoplasmic reticulum. And in from the endoplasmic reticulum, since it does not have any kind of nucleocapsid, there is no viral budding occurs in this phase at Ehrlich compartment. So this, as soon as the spike protein forms, it then moves to the Golgi apparatus, or the Golgi body, via the cis, medial, and trans. Here also the same thing happens for like manostreaming, branching, and elongation, further modification of N-glycosylation and fucosylation silation in the transglycolation, and parallelly the O-glycosylation also continues to occur. And in furin, there comes the another difference that occurs, is that when the cleavage of this spike protein with the that is produced with the help of virus occurs, there is no uh, attachment now present between S1 and S2. Because of this, S1s and S2 site are more exposed for to the enzyme for further glycosylation. And because of this, there is now difference in the glycosylation pattern occurs between uh, SARS-CoV-2 virus and the spike protein, and the spike protein produced by the vaccines. After this furin cleavage, these spike proteins, uh, S1 shedding and S1 shedding, the S1 and S2 now get separated, and there is no non-covalent attachment occurs. Because of this, O-glycosylation increased in the S2 part of the domain, and S1 part now moves to the extracellular matrix, and some part of the protein, the S1 and S2 that are not been able to cleave by furin, are attached to the plasma membrane. And and so this is a general differences, or you can say how the uh, actual virus and the spike proteins, or the vaccine candidate spike proteins that are present in the vaccine candidates process in the host cell. Thank you for your time.